Hi everyone, my name is Lilly East and I'm from Clinical Pharmacology at SAREPTA. Today, together with Dr. Erica Koenig from Clinical Development, we hope to bring you a very interesting topic. At SAREPTA, both of our functions are integrated within clinical sciences, and our work focuses on advancing clinical drug candidates in rare conditions from early development to registry and ultimately patient access. The central theme of our talk today is on using model-informed drug development to guide clinical decisions, specifically for trial patients with rare conditions. Next slide, please. So I like to open our talk with our perspectives on the key challenges associated with rare disease drug development and how quantitative clinical pharmacology can be used to address these gaps. We will then present a case study highlighting our own efforts in using these quantitative approaches to guide trial mitigation strategies due to COVID-19 dose interruptions. At the conclusion of our talk, we also like to further share our views on model-informed drug development, or MIDD, its broader application and policy implications. Next slide. So over the past 3 decades, there has been significant progress in orphan drug development. But the overall ratio of approvals to orphan drug designation, as illustrated on the right plot, remains suboptimal. Now factoring in that in the US and Europe alone, over 60 million individuals are suffering from 700 different types of rare diseases, with the majority affecting children in their development years. I think we can all agree that there's a desperate need for effective therapies for this very underrepresented patient population. Along with patients and families, those of us who work in this clinical space are keenly aware that there are unique challenges and scientific gaps associated with orphan drug development. We have called out some of the key ones that we are very well aware of on the left side. But fortunately, in the recent year, there has been a paradigm shift in orphan drug research and development. Next slide. One notable area of development is the application of quantitative clinical pharmacology in rare diseases. And this is where I think the scientific opportunities meet the demands of orphan drug development. I often like to explain QCP, or quantitative clin pharm, as an evolved version of traditional clin pharm, which was very dose-centric. But now it has evolved to an interdisciplinary field that utilizes multi-source data to inform key R&D milestones, not just the dose. I won't be able to review all of these elements on the left side, but suffice to say that the execution of this is multifaceted. And when successfully implemented, it can drive optimization and acceleration of R&D progression, contribute to the overall evidence of drug effectiveness and safety, and facilitate extrapolation beyond the patient population study in the pivotal trials. So these 3 aspects that are highlighted on the right are especially important for orphan drug development because optimizing trial design and reducing the number of clinical studies are vital in a limited patient population. Further to that point, as you can imagine, expediting the overall R&D timeline means faster patient access. A critical component of this multifaceted approach is the data integration itself. To do that, we leverage analytical tools such as the MIDD approach, which I will talk more about on the next slide. Next slide, please. So this concept of MIDD is really using quantitative models to guide and facilitate decision-making in drug development. The most prevalent techniques are illustrated on this slide, and essentially they integrate the totality of our understanding on pharmacology, physiology, and disease from both preclinical and clinical settings. These not only help us understand our own drug performance, but also make predictions for broader applications. So the value proposition of MIDD is well recognized, but its application in drug approval is only possible with a supportive regulatory framework. That said, MIDD was formally recognized and included in the PDUFA reauthorization from the FDA in 2018. So ultimately, all of these efforts combined lead to a common objective, which is efficient and effective drug development. And this is especially important for rare disease area. So now with that introduction, I'm going to pause and hand over to Dr. Koenig, who will provide an overview of our own efforts at Sarepta in orphan drug development.
Thank you. Next slide, please. So I think at first, as Lilly had explained, of using this modeling, just to give you an overview, of course, of our pipeline for Duchenne muscular dystrophy. So we have built an expansive pipeline, as you can see here, dedicated to DMD with the goal of treating 100% of individuals with the disease. And to meet this goal of reaching 100% of Duchenne patients, we have three diverse approaches to Duchenne drug development that you can see on the left: RNA targeted therapies, gene therapy, and gene editing. The purple bars do reflect programs that were developed internally at Sarepta, and then the blue and yellow bars indicate programs that are in partnership with external collaborations. For today's talk, we're going to focus specifically on our RNA program, specifically to the PMO clinical trials. In next slide, you can see that just highlighted in the red box. Next slide, please. So what causes DMD? Just to give you a brief, overview around it. So a mutation or error in the gene for dystrophin is an important protein involved in the function of muscle cells, and it is the underlying cause of DMD. As you can see in the lower left corner, using exon skipping, which is what we use within our PMOs, as a potential treatment for DMD. The most important thing to see in the schematic is how some DMD patients are missing part, they have a mutation, where they're missing part of the instructions for making the dystrophin. This mutation, as you can see with the white gap, disrupts the open reading frame in the dystrophin mRNA, and it results in a premature termination of translation. With this complete loss of dystrophin, it leads to major destabilization of the sarcolemma and weakness of the muscle fibers that undergo constant damage under stress. So what happens is, of course, as you see with that premature in the diagram, you can't make the rest of the protein and there's no functional dystrophin protein. In the right figure, what you can see, and this is how the mechanism of action of our PMOs, is when you have exon skipping, it can bypass these missing instructions. With the exon skipping, you can restore the mRNA reading frame and potentially allow for this production, as you see below, of this shorter functional dystrophin protein. Next slide, please. This is just an overview of our three global PMO programs that Lilly will go into a little more information looking at the clinical trial simulations. But within these three programs, addressing up to 30% of individuals who have DMD. Not going to go into much detail, but just wanted to point out within our mission study, these are for patients who are amenable to skipping of exon 51. And within our ongoing ESSENCE study, PMOs for patients that are amenable to exon 53, as well as exon 45. Next slide. I will now pass it back over to Dr. East to talk about the impact of the mixing doses in these trials.
Thank you. So as many of you gathered from the earlier session this morning, the COVID-19 pandemic has really posed significant challenges to all trial conduct and individuals involved in these studies. So the case study that we're about to go into is really a snippet of our organization's larger effort in mitigating the risk and impact to our patients. It also showcases an example of what I just mentioned before, how a MIDD approach can help inform clinical decision-making. This pyramid here encapsulates our overall strategy to address the impact of dose interruption as a result of social distancing due to COVID-19. Now, we proposed a tier-based approach to enable incorporating new data as they become available. Our first step, we issued a guidance to provide a qualitative assessment to the dose interruption impact and recommendations for patients receiving treatment in the commercial space. Step two is a refinement of that assessment using a quantitative or modeling framework to predict the impact on trial efficacy endpoints under various clinically relevant scenarios. The information was then used to inform trial mitigation planning in step 3. Next slide, please. So to enable a quantitative assessment, we developed a preliminary mechanistic model that integrates clinical, preclinical data, and pediatric muscle physiology. And this preliminary model allows us to predict drug concentration in the blood at the target muscle tissues and the biological endpoints that are being collected and assessed in our clinical trials. We're also extending this modeling framework across our DMD PMO pipeline as they share similar drug PK characteristics, as well as mechanism of action on exon skipping. Next slide, please. So to capture the clinical implications of the COVID-19 disruption in our evaluation, our key considerations were placed on treatment duration, dose interruption duration, prediction of clinical endpoints, and patient demographics. We assessed 3 treatment durations: 96 weeks as chronic, 48 weeks as intermediate, and 4 weeks as short treatment duration. And this essentially captures the scenarios in the ongoing trials and in the commercial space. We also used 12 weeks of dose interruption in our assumption as our worst case scenario. The level of impact from the dose interruption was then compared to the relative reference point, which is, without any dose interruption. So today, while we're not able to divulge in the technical details of the trial predictions, because these are ongoing studies, these assessments that we have concluded on: 1, magnitude of change in tissue drug concentration and clinical endpoints; 2, the dosing duration needed to compensate for the impact of dose interruption. Both of these elements helped guide the clinical trial mitigation planning, which you'll hear more about from Dr. Koenig. Next slide, please.
Thank you. So I think that Kristen and Kim, did a very good job speaking kind of around, especially Kristen in the first talk, around the guidance that the FDA provided. And they were very quick, as Kristen had mentioned, to getting this out. This is, of course, just an updated as of December 4th. And we've used this, religiously, I have to say, when really dealing with the impact on our PMO trials. And something that for us has been, we didn't go into detail, but within our clinical trials, we do weekly injections. So we do have, of course, the patients are highly impacted, and also being within DMD, being an immunocompromised population, it's even harder, of course, because of that. But we do, of course, recognize, the FDA did recognize at the very beginning this impact. And that impact, of course, is, and these are just some points we took out of this, it really depends on many factors. As mentioned, the underlying, disease itself, the trial design, as Lilly was kind of talking about different time points, and also what region the study is being conducted. Of course, these are global studies, so they're done around the world. And then again, within this, as we all know, challenges do arise. For example, quarantine, sites are closed. We have travel limitations. I think Kim talked a lot about this using an app. Having these remote assessments is very important. There's also interruptions to supply chain for the investigational product. And then we also have to take into consideration during COVID, with site personnel or even the trial participants, of course, that are becoming infected with the virus. The FDA also, recognized the fact that challenges do lead to difficulties in meeting these protocol-specified procedures. That is, of course, including administering or using the IP, the investigational product. And then also within that, adhering to the mandated visits, you have a lot of laboratory diagnostic testing. So these are all things that we really had to take into consideration during this time. And again, within the guidance, the FDA recognizing that modifications may be required. And sometimes there are protocol deviations that we just cannot avoid due to COVID-19. And throughout all of this, and when doing the simulations and using modeling in our approach, the really great thing about the guidance too is really the underlying purpose of it, to assist sponsors in ensuring the safety of these trial participants. That's the number one thing, of course, when we're thinking about this, and then maintaining compliance within good clinical practice and minimizing the risks, of course, to the trial integrity. Next slide, please. So based on what Lilly had shown and really coming together as a team and really trying to understand the mitigation strategies that we could address extending this dose interruption, and also at the end of the day, missing of those primary endpoints in our clinical trials, we had 4 options. We started with the first option, which would just be no change to the sample size. We thought about that first. And then within option 2, and that's based, of course, on what Lilly had presented, even though, of course, we didn't go into the specific data based on this being ongoing studies, but really extending that treatment duration based on the dose interruption. For our PMO trials to date, it was not recommended based on the duration of extension, and that was determined, of course, by Dr. East's work with the PK/PD modeling. And we really took that into consideration, just thinking about the length of time of some of these trials also. Option 3, what we thought within our clinical trials was doing an interim analysis for the reassessment of sample size. That was something that we could use as a mitigation within that dose interruption. But then the 4th option, which is the one that we ended up going with, is really to increase sample size. So by increasing the patients who are enrolling, we can maintain the study power to account for that diluted treatment effect that we might have within that dose interruption. And then we can bring this back with the PK/PD modeling. We can extend that to evaluate the treatment effect for following the sample size increase. So using, of course, as mentioned, the modeling and really a team effort of understanding this diluted treatment effect and everything, we were able to apply Dr. East's approach into our clinical trials. Next slide, please. So the key takeaways of the information or the talk that we just gave is really broken down into 2: outcomes and then broader application, as Dr. East had said earlier on. So within the outcomes of what we spoke about today, a novel mechanistic PK/PD modeling was developed for our DMD exon skipping therapy. So it's important to note that this can be extended across the PMO platform to project other clinical study endpoints and treatment outcomes on efficacy biomarkers. Consistent with the MIDD framework, modeling can be used to guide treatment recommendations for patients and also inform emergent clinical trial designs, and then also showing a mitigation to the program risk, which is following the dose interruption due to COVID-19. We also believe that utilizing this approach can guide our clinical and regulatory strategies to, of course, as our number one concern, or what we need to do is to ensure the safety of our trial participants and also maximize the probability of the success of the clinical trial. From a broader application, we can use evidence-based mitigation measures, informing by using quantitative tools such as the MIDD, which is recommended to mitigate these emergent risks to our trial patients. We can maintain data integrity and, of course, optimize the clinical trial success. This is extremely important in orphan drug development, where we all know there's limited trial patient population. And then finally, this initiative was possible, as Dr. East had mentioned earlier, through the FDA's COVID regulatory guidance as well as PDUFA, which does allow this flexibility to amend the protocol in response to a crisis in driving that application of using MIDD in a clinical decision making. Again, to ensure the long-term viability of this practice, FDA must be appropriately resourced to enable this dialogue and to prioritize the continuation of using this MIDD approach. Next slide, please. And then finally, we just want to acknowledge the individuals and the families who participated in the clinical trials, and also to the contributors and associates in each of these studies, as well as to our development teams, and also, most importantly, to the patient advocacy groups.